K ATPase series in Anatomy and Physiology. Chapter 2. Cell Structure and Function. Episode 7. Cell Transport. Part 2. Active Transport. If you enjoyed the video, don't forget to hit the like button and share it, to help us reach every medical and health professional student around the world. And of course, make sure to subscribe to the channel for more valuable content. Your comments are highly appreciated. In the previous session, we have observed the different mechanisms of passive transport and their roles in the cell. In today's session, we will discuss the process of active transport, a cell's special delivery system in the cell. Unlike passive transport, where substances move freely from areas of high concentration to low concentration, active transport moves substances against this flow, from areas of low concentration to high concentration. It's like pushing a boulder uphill, where huge effort and a lot of energy are needed. In the same manner, this uphill active transport is not spontaneous. In fact, it depends on specific transporter proteins embedded in the cell membrane. This transporter always requires energy to push the ions against their concentration gradient. Let's look at this short animation of the active transport mechanism. You can see that the green molecules are moving from the inside of the cell to outside, from low concentration to a higher concentration. You can also see here that the transport needs both a transporter protein and energy. Without energy, this process simply wouldn't occur. So why do cells go to all this trouble? The answer is that because they can't just let substances drift in and out randomly. Sometimes, a cell needs to import essential nutrients or ions, even when these are already present in higher amounts inside it. Other times, it needs to expel ions, wastes or toxins, even if those are more concentrated outside it. This is where active transport becomes essential. Without it, cells wouldn't be able to maintain internal balance or perform critical functions, like nerve signaling, muscle contraction, or absorption of essential nutrients like glucose and amino acids. Nonetheless, active transport comes in two main types: primary and secondary, depending on the source of energy and how it is used. In primary active transport, ATP that is normally produced by mitochondria is used directly by the transporter proteins. These proteins, often called pumps, break down ATP into other molecules like ADP and inorganic phosphate, to release energy. The released energy is then used to change the shape of the pump, which then forces ions or molecules across the membrane, against their natural gradient. One of the most well-known examples is the sodium-potassium pump. In resting cells, sodium ions are more concentrated outside the cell, while potassium ions are more concentrated inside. This pump pushes 3 sodium ions from inside to outside the cell toward their higher concentration. At the same time, it brings 2 potassium ions from outside the cell to inside. This transport mechanisms of both sodium and potassium requires one molecule of ATP as source of energy. Therefore, the sodium-potassium pump not only maintains the proper ion concentrations but also helps create an electrical charge difference across the membrane. By moving 3 positive ions outside the cell and only 2 positive ions inside, this pump plays a key role in establishing the resting membrane potential in nerve and muscle cells. Without this pump, basic cellular and bodily functions would collapse. Now, here's something interesting. As ions like sodium are pumped across the membrane using ATP, they start to accumulate outside the membrane where their natural concentration is higher than inside. This creates a combination of concentration difference and electrical charge on both sides of the membrane, which is known as electrochemical gradient. Generally, this buildup and accumulation of sodium outside the cell creates a form of stored energy, known as the potential energy. Now, sodium ions naturally want to flow back to inside the cell by diffusion. When they do that, the stored energy is released. This released energy can be then harnessed to power other processes, such as being the major driving energy for secondary active transport, as we will see in the coming few seconds. To simplify this, imagine it like water held back by a dam. When the dam is closed, the water accumulates behind the gates, creating stored energy behind the wall. If the dam is opened, the water rushes through the gates by the action of the gravity, and can be used to spin turbines and generate electricity. This is how hydroelectric power works to generate electricity. The second type of active transport is the secondary active transport. Like primary active transport, secondary active transport also transports large substance such as amino acids and simple sugars against their concentration gradient, from low to high concentrations. However, unlike primary active transport, which uses ATP from cells directly at the transporter site, the transporter protein of the secondary active transport doesn't use ATP directly. Instead, it uses the stored potential energy created by the active transport as ions flow back down their gradients by diffusion. So, secondary active transport is associated with active transport as its source of energy. In all species, secondary active transport can occur in two forms. These are symport and antiport secondary active transport systems. The symport transport is a co-transport mechanisms that move substances across the cell membrane in the same direction of ion flow. Remember that ions flow down their concentration gradient and the substances flow against their concentration gradient. In contrast, antiport or counter-transport involves two substances moving in opposite directions. The ion moves in one direction from high to low concentration, while the other substance is pushed in the other direction against its gradient. A classic example of symport transport in eukaryotes is the sodium-glucose transporter in the intestines. First, the sodium-potassium pump uses ATP directly to move sodium out of the cell by primary active transport. As we said before, this builds up a high concentration of sodium outside the membrane and stores potential energy. Sodium now diffuses back inside from high concentration to low concentration areas. The transporter protein uses the released potential energy from the flow back of sodium to pull glucose inside the cell from low to high concentration. As you see, this happens even glucose is already more concentrated inside. This is how your body efficiently absorbs glucose from food after eating. On the other hand, a good example of antiport active transport is the sodium-calcium exchanger in heart cells, which restores intracellular calcium levels after myocyte contraction. This exchanger also uses potential energy of sodium back in flow after active transport to remove calcium out of the cardiomyocyte. Interestingly, the sodium-calcium exchanger is a target therapy in patients with heart failure to increase contractility by increasing calcium levels. For example, the drug digitalis inhibits the sodium-potassium pump, leading to reduced sodium levels outside the cell. As sodium levels outside the cell become significantly reduced, this inhibits the sodium-calcium exchanger and increases intracellular calcium levels and so contractility of the heart.